All right, so we're going to talk about WA3 here. So in WA3, what you will do, keep in mind that the rubric here discusses what you're trying to get out of this, what you're trying to achieve in WA3. Really, what you're trying to achieve is, as it says here, you're writing a draft for WA5, your final version. And so just to keep you in mind about kind of what it should look like, what my expectations are, go back here to the original assignment example I gave you and model it from this one. All right, this is a great example of what I'm expecting. So if you haven't seen this, go back and look at that. I gave that to you like the first or second week. Now, WA3 will contain most of the sections of WA5. And so look here, introduction, materials, and methods. That's 1, 2, results, 3, discussion, 4, references, 5, figures and tables, 6. So you should have enough by this point to start filling in the some information, at least begin writing a paragraph or two, or hopefully more than that, for each of these sections. All right. So, like I said, it should serve as a solid foundation for WA5, so that you don't have, too much to do at the end. So, write WA3 as if the material and data will be the final version. All right, and that way, when you turn it in, you can check it, or I can check it, and you basically will see what you need for WA5, what you need to change. So it's kind of like it's letting the person who's going to grade it look at it and tell you what's wrong with it. Now, yeah, you might not make as good a grade on WA3 because, it's not the final version, whatever. But realize the one that counts the most points by and far is WA5. That's the one you're trying to get WA5 to be as close. Use this, all right? Use this as a way to find out, what you need to, what you understand and what you don't understand. You may think you understand things, it happens to me a lot. I understand. You think but then once you get it done and you get the grade back, you realize, oh, yeah, I didn't understand that. This is a way for you to make sure you find that out. All right, so let's move on down here and let's look at what we have to work with. So here's our introduction. You need to include a broad overview of Drosophila melanogaster, all right, as a model organism. So you have to go find out what makes it a good model organism, all right, emphasizing the particular biological processes involved with your mutation or gene function. All right, so you need to go find out the biochemical, genetic processes that causes that gene to change the phenotype or produce the phenotype of your organism. All right, and so of course when that mutates, when that gene changes, that phenotype is going to change because this particular process was altered. So you should have already kind of investigated that, I would hope. You may not have written much about it, but you should have investigated it. All right, you're going to introduce candidate genes, how it was discovered, or how a possible mutant was identified or isolated. Discuss at least 4 other related genes. All right, elaborate on the basic biological and molecular processes involved with the mutant gene function. All right, so it's a mutant, so it's gonna work right. Explain that. Much of the introduction could be used in WA5 with very few revisions. So the idea is this introduction, at the very least, should be, ready to go for WA5. So you kind of want to, this one, you're going to pretty much put in all the information, hopefully, that you need for WA5 here at WA3. All right, you want to kind of get this part done and get it out of the way. It's just the introduction. Now, materials and methods. Well, in materials and methods, right here, describe how they were manipulated. All right, how, in other words, in our particular case, it's the software. In the past, this has been a pretty long section because people had to talk about how they grew the flies, how they operated in the lab, and how everything worked. Well, now you have to explain the software. what you crossed. Explain, and that's exactly what we're saying here. Explain how the crosses were conducted. All right. So you need to talk about the website, the software, how the crosses, how you actually did your crosses. Explain how the phenotypes were scored and discuss how the statistical analysis were performed. In other words, what did we do? We added the numbers up that we got from FlyLab, and we did chi-square. We did some other things. You did the mapping, all of those things. Whatever math you did, whatever statistical analysis you did, you need to put that in here. Now describe how relevant equipment and materials were used. Again, this is FlyLab. In the past, this has been, how did the equipment work? What equipment did we use? That sort of thing. But, what kind of food did we feed the flies? Well, here it's just going back to the software. So really, this is almost identical to this. All right. This one also should be completed, or be really close to being completed, your final version at the end of WA3. If you make a few mistakes here and there, won't be taken off too much. And if it is, well, good. You get that fixed and ready for WA5. All right, now we move on to our results. So describe the genetic basis for what you've done. All right, basically your outcomes, put the graphics in there. This has kind of been done already. If you didn't do chromosomal charts, I suggested that you do them for the last one, and a lot of you did. So do the chromosome charts. If you don't know what I'm talking about, make sure you do the mapping, so you show that the crossing over, etc. Of course, the mapping charts, not the same. Well, chromosomal charts either way. Something showing the chromosome is a chromosomal chart. So we kind of have two different types. One is a mapping, and the other one is just illustrating the crossing over of your crosses. So make sure you get all of those in here. These should go in your results. Put in the genetic basis for the rationale for your allelic cross, your mapping cross. Describe the outcome of your crosses. All right. What are your results? What did your numbers say? Mainly your mapping. The mapping is the important part. Let's see. Brief discussion and conclusion of what the data suggests. This could serve as the basis for discussion. So this is maybe the hard one, and the one you're going to probably leave a little for WA5. But at least start it. Put in a brief discussion and conclusion about what the data suggests. What do the results suggest? Do they suggest what? The location of your gene? You may also at this point have some results from the bioinformatics that suggest what type of mutation you have. Where were the changes in the nucleic acids? Did it splice different perhaps, or is there just like one letter change and it caused one amino acid to change, and could that really make this big a difference? Well, sometimes yes it does, and you'll have to investigate that, find out if these changes in the nucleic acid. So at least begin putting that type of information down. So the discussion, there should be a discussion. You need to at least start it. I'm not going to say a certain length. If you want to make sure that you do enough, try to complete it. Put down everything you know, but you can't put down information you don't have yet, which means you can't do a lot of the bioinformatics stuff yet. But you can at least start it. You can certainly put down the discussion and what the data suggests from your mapping. So the location you can discuss. The mutations you might not be able to discuss very much yet. All right, so list the references. Actually, everybody did really well on the references. I mean, there were a few who didn't, but by and far, the majority of you have references. You'll need some more. Of course, we're going to be talking about more things here. Specifically, I think the biochemical stuff is a good place to where you'll have to reference what you talk about. And you should probably put that, add the biochemical pathways, talk about the functioning of the proteins, what they do, how they chemically work within the cells, that sort of thing. And of course, you'll need references for those. And remember to paraphrase. Do not copy directly from any article. You'll get dinged for plagiarism on that. So just make for sure you've got that, and get 6 to 10, maybe more. 6 to 10 is the minimum of 6. 10 at this point. I won't count off if you put more. So get those in there though. They need to be there at least 6 to 10. All right, at least 6, preferably 10. That's the way I will say it. Now the figures, actually most of you did really well on the figures. Maybe some minor things you left off. You got a point here or there, but make sure to include a legend. And I would say look over here at where your points are coming from on this one. This is where you're going to, okay, why did I miss this little thing and it counted so much? Well, look over here. What counts the most? Well, at this point, it's the results. All right, so we don't have all our results. We don't have the bioinformatics, or not much of them, of the bioinformatic results, but you have your mapping. So realize WA2 goes in here, use that, get your points. All right. The discussion is 20. All right, so that should be relatively easy. You can only discuss what you have here. You can't discuss more than what you have. So that should be an easy 20 because you can't do too much here, and it counts a lot. So make sure what you do put here is good. Figures and legends, 25. So this is, most of you were doing really well on your figures and legends. I didn't have a problem with it. Make for sure you go back and check this paper. Look how they did it. Let's go scroll down here. They don't miss much here in this paper. Let's find their, it's going to be at the end, of course. Well, if my system will cooperate. Looks like it locked up. It is. Notice how long their paper was, 61 pages. Yours. Let's see if we a lot of this. Guys, and legend. Huh. Yeah. All right. How this looks. This is exactly. It's a little short. Maybe they should have said something else, but this it needed a figure 1, a title, and then description of what's going on up here. Some type of description that talks about, that gets the idea across as to what's going on in the picture. You can't just say A is this, B is that. You have to say A is this showing something, B is this showing something else. That really needs to be how you put that in there. All right, that also shows you how to do. Yeah. So take a look at this paper, especially that I've given you. Work from that one. All right. So take a look at this. All right, and it's going to be in the Dropbox in the morning. So check it out, and if you have questions, we are going to talk later in the week. And remember, you can talk to me personally if you want to Zoom. Email me, talk to me personally, Zoom with me. Most days I'm available at least sometime during the day. And I have talked with several of you. But if you don't have any questions, my office hours are wide open for you, is what I'm telling you. All right.